Folks, here is a quick lesson on ethology to begin our lab. Ethology is the study of animal behavior, and this means any animal, right? A lot of people ask why study an animal's behavior. There's a lot of reasons, but for me personally as an environmental scientist, the reason to study animal behavior is to see if things we're doing to the environment is affecting their behavior. We don't know what normal is unless we study those behaviors and find out what normal is, so that in the future, if things change, we can figure out that something has changed. So if we're studying a species every year and seeing what their normal behaviors are, then we can notice when there's a change in that behavior. And then we can make correlations as to what that change, what that stimulus that changed that behavior is. The problem with animal behaviors, though, is trying to objectively document and measure those behaviors. A lot of people try to anthropomorphize and go, oh, the dog is sad. We don't want to do that. We want to record behaviors objectively. So I'll show you. First, behaviors can be innate or learned. Innate behaviors are behaviors you're born with. These are hereditary behaviors. Classic mammal hereditary behavior is the ability to suckle. Every mammal is born understanding how to do this, how to get milk from mom. Learned behaviors are the ones that you're taught throughout your lifetime, or that you acquire through experience throughout your lifetime. For humans, this is a lot of things, but generally animals learn not to bother a porcupine or not to bother a skunk. And they're not taught that, they just learn that, and that's a new behavior that they're going to learn. The things that elicit behaviors, so when we're biologists trying to study these behaviors objectively, the things that elicit behaviors are our senses. So we break them into 4 categories. We have auditory, visual, tactile, and chemical. So those are basically the 5 senses, but for a biologist, both scent and smell is chemical. We don't look at them differently. And then obviously being able to see something, be able to hear something, and being like actually touch something or something touch you. This is probably the most important history lesson I can give you in ethology. And these are Tinbergen's questions. So he always asked how and why. How is a behavior happening? What has caused it? What are like the physical mechanisms that make that behavior happen? So we can look at this in the moment, what is causing it, but also genetically. How did this evolve? Why? Why did it evolve? What is the survival value? Why does this creature have this behavior? What could happen that would allow this to evolve? And then you also can look at the phylogeny and ontogeny of behaviors. So phylogeny are the evolutionary patterns that led to that behavior. So we can look at how and why that behavior has evolved over generations. And then we can look at the ontogeny of a behavior. The how and why has that behavior evolved in a single organism's lifespan. Phylogeny and ontogeny. So back to trying to do this objectively, we as biologists, as ethologists, have created ethograms. So rather than looking at a dog's posturing and saying, ooh, they look sad, or ooh, they look happy, we can look at that posturing and record exactly all those different postures that you create. And then instead of saying this is a happy posture or this is a sad posture, we can say that this stimulus elicits this posture. And because of that, we are assuming that this is an aggressive posture because of the stimulus that we've created. I'll go through that a little bit more with some diagrams. This is the ethogram for a cuttlefish. These are all the different postures that a cuttlefish can make, and then they would record when those postures happen. So when we give them food, what postures do they do? When we poke them, what postures do they do? When there's a mate nearby, what postures do they do? And accumulating a resource of which postures happen when, then we can start correlating whether postures are aggressive or submissive. Here's more specifically facial features. Dave Hackett has done a lot of research on wolves, so these are the facial features of a wolf. And we can then record those different facial postures and correlate them to different behaviors and different stimuli. Here's my research on redback salamanders. And we look at their posturings. They're quite small, so their postures aren't as different. But we can have a salamander that's laying on its belly or raised up on all fours, and then we can specifically look how raised up are they. And this particular paper looked at how raised up the salamander is and correlated that to how aggressive they are being, that the more raised up they are, the more aggressive they are. And they did this over several studies where they put two salamanders together and figured out a dominance chart of which salamander is the most aggressive, which is the most dominant, and made correlations. We're going to look at gulls. I have supplied in the handout some gull postures. The reason we're looking at gulls is because A, they're super common. I'm hoping that all of you have access to gulls. And B, I do have access to a decent gull posturing ethogram that I think you guys will be able to use. And C, it's really easy to add a stimulus to gulls because we can just feed them crackers. It's a pretty simple thing that I don't feel super guilty about affecting the wild populations of gulls by feeding them crackers. The two gulls that you're going to see in Ontario typically this time of year are ring-billed gulls and herring gulls. The key difference here, ring-billed gulls have yellow feet and herring gulls have pink. I remember this like a red herring is a word we use, so they have pink feet. Ring-billed gulls do have a ring around their beak, but that's often harder to tell. Herring gulls are almost twice the size of ring-billed gulls, so if they're together, they're very easy to tell which is which. I've included pictures here where the ring-billed gull has some gray on its face, whereas the herring gull doesn't. That is not an indicator of species, that is an indicator of age. Young gulls, for the first 3 or 4 years of their life, will be gray and speckled. It's not until a gull reaches breeding age that it gets this beautiful white, gray, black coloring. And that doesn't last all year. That beautiful white, gray, black coloring is just for the breeding season. So in the winter, all the gulls are a little bit speckled. I've just shown two different ones here, like the ring-billed gull in this picture is probably a little younger, or it's in the winter, whereas the herring gull is in its bright breeding colors. Just to show you, because you're going to see some of everything, and I want you to be able to determine that. Because it's important that when you're testing your gulls, that they're all the same species. It doesn't really matter long term, but it would be nice if we were all using just ring-billed gulls. Herring gulls are less common, but we'll see how it goes. Use the gulls you find. Our objective for this week's experiment is we're going to look at which behaviors are greater before food and which behaviors are greater after food. And we're going to make the correlation that the birds that are more aggressive are going to be fighting for that food, that stimulus, and they're probably the more dominant of the birds. And those are correlations from years of ethology research. I've included at the end here some past data just to give you guys an idea in case you may or may not get out there into the field. But this is 1 year's class, what they came up with. I have spreadsheets online that you can use. I have handouts that you can use. Go collect some of your own data. It's a very short activity and I think rather fun.